cellular adaptation to stress, definitions, and examples. In this question, if you pick this question up in the exam, you will briefly have to talk about the adaptation responses of a cell. And I say briefly because the questions that follow in your list, questions 5, 6, 7, I believe, selectively focus on the individual processes we will be discussing in this question. So what I'll try to do is I will detail them out as much as is required for the extent of this question, but also in a way that you can really cover the content you need for the following questions as well, so you don't really have to come back to it or do it, separately. So the first thing we've got to do is actually define what adaptation is. It's always a good thing in pathology. Whenever you get a question, you always define what's going on and then you build on from there. When we get to special pathology, you guys will have a scheme of work that you'll follow. But anyways, adaptation. Adaptation in simple terms is used to describe any change that takes place in a cell as a response to a physiological or pathological stimuli. So I'm just going to write that down really quickly. Changes in a cell as a response to a pathological or a physiological stimuli. Right, and just to reiterate it one last time, stimuli, you have a stimuli. It can be endogenous, as in the case of hormones in the body that produce an excessive response to cells, or it can be exogenous due to, let's say, for example, something as simple as just lifting weights or something that causes muscle changes in your muscle mass, right? So stimuli can be both physiological, it can be pathological, and it can be endogenous or exogenous. So let's discuss each of the individual types of cellular adaptations in a bit detail. The first one is atrophy. Now, the way I like to divide atrophy is simply hypertrophy or numerical atrophy, and these are a little different in the sense that hypertrophy can be also defined as just simple atrophy, where you have tissue degeneration secondary to a decrease in protein synthesis and an overall decrease in cell content, right? And numerical atrophy is a reduction in cell number. The name gives it away. And the mechanism behind this is secondary to degradation rather of your proteins within the cell, and this is achieved through your ubiquitin and proteasome pathway. And normally, if you have a look at this under a microscope, under electron microscope, you will see that there are an increased number of autophagosomes. In the previous questions, you would have covered the different types of cell deaths, and so autophagosomes is something that you should be familiar with and what they really are and what they do. Not really going to go into too much details of that. So again, in simple terms, atrophy is divided into two different types, hypertrophy and numerical atrophy. And the definitions are that in simple atrophy, you have tissue degeneration caused by a decrease in cell synthesis and cell content as a whole, whereas in numerical atrophy, you have a reduction in the number of cells, right? Some examples that we need to talk about which are relevant for the exam. First type is a physiological one, so I will write that down, and it has to do with the thymus gland. And as you know, the thymus undergoes degradation in people as they get older. By puberty, that's pretty much gone. So the term that we use to describe this sort of atrophy is involution of the organ, which basically means that the organs are temporarily enlarged and then undergo degradation via an atrophic process. Simple as that. This holds true for the thymus, and another example that you can think of where it holds true is also the breasts, for example, in females. Another type of physiological response has to do with what we call senile atrophy. And the reason I've really put in this microscopic picture for you is because I want to focus on a specific pigment that deposits into tissues as a whole when people age and as part of senile atrophy, and this is known as lipofuscin. So with aging, as part of the aging process, you will see that within organs, and if you observe them under the microscope or when you observe them more often than not in an autopsy in old people, you will see that there is a yellow to brown granular pigment found deposited in these organs. And what I will do is that because this is an important morphology, I will highlight this in yellow so you remember this. And the other thing to remember in senile atrophy is that it's basically associated with your regular wear and tear that occurs as a process of aging. And you don't really need to know too much about lipofuscin for this question, but there is a question about it in pigments, I believe. There's a rather part of it in the question of pigments, I believe, and you will explore this in more detail over there. So I will just leave it at this for now. Right, and the last type of atrophy we're going to cover are the pathological ones. And these pathological ones can be divided into generalized or local, and also in some cases, in some textbooks, you will see neurological as well. What is the basic idea? In generalized atrophy is attributed to a state of high catabolism in the body. So this can be in the cases of, let's say, malnutrition, or in the cases of cancers, where muscles are broken down to as a source of energy, providing a fuel for energy. Simple as that, right? Localized atrophy has to do with muscles as well, and this can be due to, I don't have space over here, so I'm just going to write it up above here. It can be associated with inactivity. It can be associated with pressure. Like pressure induction too can cause atrophy, and in some cases it also has to do with loss of hormonal stimulation. So any decrease in hormonal stimulation can also result in atrophy. An example of this, if you are asked, you can just really easily mention osteoporosis, right? And then neurological atrophy, the word really gives it away, but it is indeed muscular atrophy as well, occurring secondary to a loss in neurological stimulation. And so that covers what you should be able to talk about for atrophy with some examples. Let's move on to hypertrophy. Now, hypertrophy, by definition, is an increase in the size of parenchymal cells leading to an increase in the size of the organ. I think it's quite easy. The word really gives it away, but writing it down just here is that there is an increased size of parenchymal cells that lead to an increase in organ size. It's really as simple as that. Now, the causes of an organ undergoing hypertrophy could simply be because they have an increased functional demand. So increased functionality of that organ can be a cause of hypertrophy, and it can also be caused by stimulation, by hormones and growth factors. So those are the causes, and mechanisms by which hypertrophy occurs is just an increased production of cellular proteins secondary to these hormones and growth factors, or these stress factors, resulting in an increased demand of the organ's functionality, causing an overall increase in the production of cellular proteins, and you basically have subcellular organelles undergoing selective hypertrophy. And that is pretty much it. That is how hypertrophy is caused. Some examples that you should be able to talk about pathologically, so I will just write pathological here, is heart or myocardial hypertrophy. When we talk a little bit about heart failure and the circulatory part of general pathology that covers circulation, we will discuss the different types of hypertrophy the heart undergoes. There are two different types in particular, if you want to know for your knowledge for now. You've got eccentric hypertrophy and you've got concentric hypertrophy, right? And the one I really wanted to focus on for now is concentric hypertrophy, because this is what occurs secondary to hypertension or secondary to aortic stenosis. And these are two other important pathologies that you will cover when we move to special pathology. But for now, you're good to know that myocardial hypertrophy occurs secondary to hypertension and aortic stenosis. Hypertrophy can also be physiological. You can have your uterus enlargement due to hormonal changes as they occur in the menstrual cycle. And as I mentioned quite earlier at the start of the video, it can also be just increased muscle mass because you're athletic, you go to the gym, you're sporty, whatever. So that is something too that falls under the category of hypertrophy, just that it is a physiological response and not a pathological response. Right, with that done, we move on to hyperplasia, which is by definition a controlled proliferation in the form of elevated reproduction rate of stem cells and differentiated tissue. And I mean, I really just ended up telling you the mechanism for the most parts, but having said that, basically, by definition, hyperplasia is just an increase in the number of cells in the organ at hand that we're discussing. Please do not confuse hypertrophy and hyperplasia. They're two very different situations, and you will most likely be penalized if you make this mistake in the exam. So remember, hyperplasia has to do with an increase in the number of cells, whereas hypertrophy has to do with an increased size of said cells. The mechanism for the most parts are pretty much the same as hypertrophy, is that, and in this sense, you just have proliferation that is induced by growth factors and resulting in an overall increased output of new cells from stem cells. Now, we're going to talk a bit about the examples. And in this diagram of the uterus and the endometrium in specific, which we're going to be focusing on, so I will just write endometrium here. You can have both a physiological and a pathological response resulting in hyperplasia depending on the amount of stimulation you get from estrogen, right? So physiological hyperplasia, estrogen stimulation of the endometrium in the menstrual cycle. But pathologically, if you have excess stimulation by estrogen, this can lead to endometrial hyperplasia. And this endometrial hyperplasia, when you guys get to female reproductive pathology, you will see that this endometrial hyperplasia secondary to an excess stimulation by estrogen can serve as a precancerous lesion. I will just leave it at that for now. Don't really need to get into too much details just yet. You will learn it as part of special pathology. Right. Another example that is physiological hyperplasia is that I want you to imagine someone who has hemolytic anemia, right? As a compensatory mechanism, you will have a reactive bone marrow hyperplasia. And yeah, so this is a physiological response. The pathology of hemolytic anemia, the pathology at hand here is hemolytic anemia, but a physiological response to a decreased red blood cell count is a reactive bone marrow hyperplasia, right? And another pathological one, good to know, again, you will cover it when you move on to male reproductive pathology in more detail, but for now, benign prostatic hyperplasia. This is an increase in the number of cells of both your stroma and your glandular cells of the prostate, and this occurs in some cases. It's not, I wouldn't really say it's a pathology as such, because it is quite normal and it does occur in males that are greater than 50 years of age, but for the most part you can classify it as a pathology, but it is that it is a normal response as well in that sense, right? It occurs to everyone, but it's not something that is, physiological. Is that you shouldn't really be having an enlarged prostate to begin with. But yeah, that should really be it. A really important thing I'll just mention now itself is that this benign prostatic hyperplasia has absolutely no risk of malignancy. And I'm only mentioning this because we did discuss it when we discussed endometrial hyperplasia that it can be a precancerous lesion. In the case of benign prostatic hyperplasia, absolutely no chances of malignancy or prostate adenocarcinoma. Okay, so before we really finish off hyperplasia, we need to discuss different types of cells and different types of tissues, right? And the three major ones at concern here are labile tissue, permanent tissue, and stable tissue. I will discuss this very briefly because you will talk a lot more about it in regeneration. I will cover that very briefly as well, just to give you an overall picture of why this is important. Right now, labile tissue is tissue that is relatively short-lived, and it is rapidly dividing. And this rapid division is primarily due to an established stem cell population. stem cell population. And so these labile tissues, you can imagine hyperplasia would only take place in tissues that are able to rapidly divide, because it is a type of controlled proliferation. And this is why it's important to mention what type of tissue can actually undergo hyperplasia as compared to tissues that cannot undergo hyperplasia at all, just because they do not have the established stem cells that are able to increase cell population. So examples of labile tissue, I will write it up here because I ran out of space at the bottom. An example is your mucous membranes of the GIT, right? They are very much capable of dividing. Same goes with the respiratory tract. And another important example, since we discussed it as well, is your hematopoietic tissue. So these are important, right? Permanent tissue have absolutely no capability to replicate throughout life. And if they cannot really replicate, if they cannot really proliferate, it basically means that these tissues, the cells of these tissue cannot undergo hyperplasia, right? Examples would be your cardiac muscle cells. And again, please do not confuse this with hypertrophy. I did say the cardiac muscle cell can undergo hypertrophy, but hyperplasia, not so, right? And another example is probably just your neurons, right? Then we have stable tissue, right? And now stable tissue, the reason I included this cycle, the cell cycle here, is that, you know, once a cell undergoes mitosis, it does have the option to go into the GO or G0 phase, whatever you want to call it, right? And that a cell can reversibly then enter the G1 phase as well once it's ready to divide. Now, these stable cells are normally the ones that exist in the GO phase of the cell cycle, but when stimulated, they can undergo cell division as well. And as such, even these cells then will be able to, these cells will also be able to undergo hyperplasia, right? And examples of, so like stable tissue is basically expandable tissue. That's how I like to think about it. That they exist in G0, but can progress to G1 if and when necessary. And so examples that you can write for this is just your connective tissue for the most part, the liver, and renal parenchyma. You guys do know that the liver has some sort of regenerative capabilities. More on that when we get to liver pathology. But just to give you an idea and just to give you an example of why this is important. Now, from the exam point of view, one important thing that you need to know, and I will put it in red for that very reason, is the difference between pathological hyperplasia and cancers, right? Pathological hyperplasia, even though it is occurring, you are having proliferations of cells, you are having an increased number of cells, this process is still controlled. And that is what is very important here, which basically means if you remove the stimuli at hand, the hyperplasia should resolve. In the case of cancers, this does not happen. Cancers have absolutely bypassed all normal regulatory control mechanisms, so they do not really care if you are removing the stimulus at hand or not. And so that is how you, at least from the functional point of view, differentiate between what is a pathological hyperplasia and what is actually a tumor or a cancer. Hope that made sense. Now, another important thing we need to discuss here, since we're already on the topic of cancers as well, is anaplasia and what this really is. In simple definition, anaplasia is the loss of mature cell differentiation. And so this means that they no longer resemble, or they no longer have morphological features of your regular mature cell. And the only real example where this is relevant, anaplasia for the most parts, is when a person or a patient has a malignant transformation. We leave it at this for now, because when we discuss tumor pathology as part of general patho, we'll talk a little bit more about anaplasia and dysplasia as a whole, but for now, just a brief overview. Right, coming to metaplasia. Now, this is a very important one, and I'm going to try making it as clear as I possibly can. By definition, what metaplasia is, and there are some very important words I need you to focus on in this. The first thing is that metaplasia is a reversible change. So that is something that is very important. I will highlight it because it's important. It has caused confusions, and we were told by the professors So we're just going to clarify it for you. It is a reversible change in which one differentiated, so this is an important thing, is that the cell that undergoes this change is fully differentiated. So one differentiated somatic cell type is totally, I mean not totally, but let's just say it's replaced by another fully differentiated cell type in the same tissue. So what you really need to pay attention to in this case is that metaplasia is a reversible process, and it occurs when you have one fully differentiated somatic cell type changing into a second type. And that is how you should define it. I would say this is probably one of the best definitions for it. Obviously, if you have any better ways to do it, by all means. But these are the things you need to focus on in your definition for what metaplasia is. And the mechanism by which this occurs is that you have reprogramming of the precursor cells that are already resident in the normal tissue. Right? And so it is these precursor cells, another important thing, it's these precursor cells that undergo differentiation into a new cell lineage. Right, and then again, the underlying mechanisms of this could be due to signals generated from cytokines, growth factors, components of your extracellular matrix, the cell's environment, all of that good stuff, right? But these are the things you need to focus on. As far as types are concerned, you can have what we call epithelial metaplasia, and you can have the second type, which is your mesenchymal metaplasia. I'm not really going to go into too much details of mesenchymal metaplasia. I will just mention a few of them right now to get it out of the way, because there's no real specific questions on these in the future questions. So you can, the two examples you can give are of osseous metaplasia, which can occur in arterial walls. It can occur in muscles as well, in which case we call it myositis ossificans. And the other type that we have in terms of mesenchymal metaplasia is cartilaginous, and this is normally seen in fracture healing. More of that when you do locomotor pathology. Right, and epithelial metaplasia, now these are the examples that you should be able to talk about, and there are a few important ones that we need to discuss. And the first one of relevant importance is Barrett's esophagus. And what is basically happening in this is when you have gastroesophageal reflux disease, when there's a lot of acid making its way up into the esophagus and irritating the esophageal lining, you have an intestinal metaplasia. And what basically happens is when this metaplasia takes place, you have an overall increased risk of adenocarcinoma. And of course, just to really quickly mention, normally in the esophagus you have squamous epithelium, which by metaplasia becomes columnar epithelium, which resembles the intestine. So that's why in some cases you may hear this being referred to as intestinal metaplasia of the esophagus. Right, another important example that we need to discuss is that in the bladder. And you will remember from microbiology, a specimen or an infectious agent, which is Schistosoma hematobium. And this, I think in microbiome, in the sketchy videos at least, it just mentioned that this increases the risk of bladder cancer. But for the case of pathology, what you need to know is that histologically what type of bladder cancer it results in. And so normally from histology, if you remember, your bladder is lined by what we call transitional epithelium, right? And what basically happens is within this transitional epithelium, metaplastic changes due to irritation by Schistosoma hematobium is changed to squamous. I believe it's squamous, yes. It's changed to squamous epithelium, and so that's a metaplasia as well. That's another example that you need to know. So squamous metaplasia of the bladder due to schistosoma infections. It can also occur secondary to catheters or urinary calculi, but for the most part, schistosoma infections are the most common cause, right? And then the last one within the bronchi is squamous metaplasia of the bronchi, which is replaced by pseudostratified columnar epithelium. Rather, sorry, it is a pseudostratified columnar epithelium that is replaced by a squamous metaplasia. And so an important point to note is that when we say squamous metaplasia, we're saying that an existing epithelium lining is being changed into a squamous lining. So the name of the metaplasia, for nomenclature, is the type of cell type that exists once the metaplastic change has taken place. So in the case of lungs, just clearing out one more time, in the case of bronchi rather, it is that you have your pseudostratified columnar epithelium changing to squamous epithelium. And then this also increases the risk of cancer in the lungs. More on that when you get to lung carcinomas. And moving on to the last type, now dysplasia, what it really is. This is not really a true adaptive response, so I will write that down here. Not a true adaptive response as such, but this is basically defined as a disordered growth of the epithelium, and it is characterized by a number of cellular changes. And since cellular changes in this case are microscopic changes that you obviously do need to know in this case, because dysplasia, as we'll just discuss in a bit, can lead to cancers, I would highly recommend you know these three specific changes that are relevant at the microscopic level for when we discuss dysplasia and cancers as a whole as well, right? And that these changes are abnormally frequent mitotic figures, which basically means you will see a lot of cells undergoing in different phases of the mitotic cycle. Pleomorphism, which basically means that the cells are completely almost different from their actual morphology. They could have many nuclei, they could have an increased nuclear to cytoplasmic ratio. That's one to mention as well, so I'll write that down: increased nuclear to cytoplasmic ratio. And then you can have clumped chromatin. And so these are basically features of what we call an atypical cell. And I would highly recommend you remember at least these few microscopic details. Normally you don't have to focus too much on microscopy, but this is something I would recommend you remember. So just a bit more about dysplasia to finish off really quickly is, if you have the underlying cause that is causing this disordered epithelial growth, if the underlying cause is decreased or removed or alleviated, mild to moderate dysplasia, and this is important, mild to moderate dysplasia that does not affect the entire epithelium, so the entire thickness of the epithelium. And I will just draw this out for you to explain what I'm getting to in just a bit. Imagine this is your epithelium, right? You've got 1, 2, 3, 4, 5, 6, whatever layers of cells in your epithelium, and this is a second one, right? Oops, again same thing, about 6, 7 layers. If the dysplastic changes that are taking place are, let's say, within the first 2 layers only, give or take, right, what basically happens is that this is what we classify as mild to moderate. And severe dysplasias, the more layers that undergo these dysplastic changes, where you have cells that are abnormal, they're pleomorphic, they have clump chromatin, so on and so forth, the more layers of cells within the epithelium that this change is seen in, the more severe the dysplasia is. So in the cases of mild to moderate dysplasia, if the underlying cause is removed, you can have a regression. But in the case of severe dysplasia, for the most part, unfortunately, oftentimes this becomes irreversible, and then this can develop into carcinoma in situ. There's a whole question on, not a whole question, but there is a question that specifically talks about dysplasia and anaplasia in the tumor series. We'll discuss more about these changes and how carcinoma in situ progresses. But for now, this should be enough. And just one last mention, which I did not mention, but you may see the word being thrown around, so I will put it in here, is dystrophy. And this is a term that is used to describe degeneration of tissue or organ, which is normally due to malnutrition or hereditary diseases, right? So I'll just write hereditary diseases here. And if you're asked to give an example, I think the best one you can really talk about is Duchenne's and Becker's muscular dystrophy. So the two different types of muscular dystrophy. You will cover this in locomotor pathology as well, but just to give you a overview of what the disease process actually is, I will draw it out over here for you. Within your muscle, let's say this is your extracellular matrix, you have a complex which communicates with the intracellular proteins you have within your muscles, right? And one of these intracellular proteins that is relevant to us is dystrophin. And so dystrophin communicating with this with the extracellular matrix through this protein complex, helps to keep your sarcolemma completely stable. And in Duchenne's and Becker's muscular dystrophy, there is a loss in functionality or just an overall loss in the actual production of this protein, and as such your sarcolemma is completely screwed. It's completely gone, right? It isn't anchored properly. It has changes in its shape, it changes in its functionality. And the biggest problem with that is that if you're affecting the membrane of any cell as a whole, muscle in this case it's the muscle, what you're doing is that you're making it very permeable to ions, and because of that it leads to cell death, and that is why you have regressive changes or degeneration of muscles. So that basically explains the entirety of this question, and I hope this helps.